Hello, my name is Vadim Karnaukhov. I'm a PhD student at Skolkovo Institute of Science and Technology in Moscow, Russia. My work is dedicated to the development of a statistical potential for prediction of T-cell receptor peptide recognition. T-cells are the key actors of the adaptive immune system. Specificity of a T-cell is determined by T-cell receptor, membrane-anchored protein, which recognizes peptides presented by special protein MHC. MHC proteins are expressed in almost all types of cells in human body, and they display peptides from degraded intracellular proteins, thus displaying intracellular state for inspection by T cells. If a T cell using its T cell receptor detects some foreign peptide presented by MHC, for example peptide from virus or bacteria or mutated protein in cancer cell, this T cell may be activated and kill cell presenting this foreign antigen. So T cells are very important for antipathogen and anti-cancer immunity. In each individual T cell, its T cell receptor is formed independently in the process called PDG recombination. So in each of us, there are more than 10 in the power of 8 different T cell clones with unique T cell receptors with different specificities. These TCRs are called repertoire. Recent advances in high throughput sequencing enabled accurate profiling of TCR repertoires. But the main question in this field is what antigens, what peptides are recognized by these TCRs. Prediction of TCR specificity is very important for a number of applications in cancer immunotherapies, in treatment of autoimmune diseases, and in vaccine design. In this work, we aim to develop a method for rapid screening of TCR specificity. That is, what peptides are recognized by the given TCR. We use structure-based approach when we take structure of TCR peptide MHC complex with some peptide, or for example, model from homology modeling, and a list of candidate peptides. Then we mutate original peptide in this structure for each of candidate peptides, and for each peptide estimate energy of interaction with TCR, and then provide a ranked list of these peptides based on their energy of interaction with TCR. The central question in this approach is what potential to use for estimation of TCR peptide energy of interaction. As we aim to develop a rapid method, these calculations should not include computationally expensive stages of structure refinement. So this potential should be robust to small inaccuracies in model structure, and this is appropriate for residue level potentials, not atomic level. And also this potential should be TCR specific, derived from statistics of amino acid contact preferences in TCR peptide interface in non-TCR peptide MHC crystal structures. And the main focus of this work is development of such a knowledge-based pairwise potential that we call TCRen. That is TCR energy. Why can't we use for this task some general approaches, some general energy functions? The reason is very special nature of TCR peptide interface compared to other protein-protein interactions. Other protein-protein interactions are mainly characterized by high affinity and are mainly mediated by non-specific hydrophobic interactions of hydrophobic patches on the surfaces of contacts in proteins. But for T cells, from biological state of view, this strategy is not appropriate because hydrophobic TCRs will be very cross-reactive and can cause autoimmune diseases. So TCR peptide interactions are mediated by mainly mediated by polar amino acids. These contacts are has lower strength than hydrophobic interactions, but more specific. And in general, TCR peptide interaction is characterized by low affinity. Another feature of TCR peptide interface is its asymmetry. Peptide contacting regions in TCR are depleted in non-polar hydrophobic amino acids, so as to reduce risk of autoimmunity, and enriched in polar amino acids. In their site, peptides may be regarded as a sample from human proteome with amino acid composition close to protein 1. So the interface is asymmetric. In this slide, a scheme of Tcrn derivation. First, we collected all TCR peptide MHC crystal structures from PCB, and then selected a non-redundant set of them, 91 structure. Then from this 91 structure, we extract all pairwise amino acid contacts and summarize them in a matrix. And then from this matrix, calculated amino acid contact preferences. We considered both asymmetrical setting, when we considered amino acid at the same time in TCR site and in peptide site as distinct entities, and in symmetrical setting, when we considered amino acid contacts without attention from which side these amino acids came. Also, we considered 3 reference states. In total, we considered 6 versions of Tcrn with different symmetry and reference state. We compared to these versions in several benchmarks that I will mention further, and the best results were for asymmetrical Tcrn with contact fraction reference state. And all results I will show you further are for this version of Tcrn. And here are formulas used for its calculation. Tcrn may be represented as a matrix with 20 rows and 20 columns, and each cell corresponds to a value of pairwise energy of interaction between the given 2 amino acids. Tcrn is very different from other pairwise potentials. In this slide, I show 2 representative examples: Miyazawa-Jerringen potential, derived from amino acid contact preferences in protein cores for protein folding, and Keshkin potential that represents amino acid contact preferences in general protein-protein interfaces. We see that Miyazawa-Jerringen and Keshkin looks rather similar, now with clear clustering of non-polar, polar, and charged amino acids, and for Tcrn we don't see such a regular pattern. But the most important question is what is performance of Tcrn, how it can be used for prediction of TCR peptide recognition. And this is the most important benchmark for this question. How Tcrn can be used for distinction between cognate peptides recognized by TCRs and unrelated peptides. We took TCR peptide MHC crystal structure and calculated energy of interaction between peptide and TCR in this structure. Then we in silico mutated peptide in this structure for sequentially for 1000 unrelated peptides that differ from the cognate epitope in all non-anchor positions. And for these unrelated peptides, we also calculated energy of interaction with TCR. And then we calculated cognate epitope rank. This is matrix that we used in this benchmark, that is the fraction of unrelated peptides that have more favorable energy of interaction with TCR than their cognate epitope. We repeat this procedure for all 91 structures in our non-redundant dataset, and here are distribution of obtained values, and we see that median value is 2%. That means that on average, only 2% of unrelated peptides have more favorable energy of interaction with TCR than their cognate epitope. That means high accuracy of a distinction between cognate and unrelated peptides using TCRen. In this slide, comparison of TCR performance and performance of other pairwise potentials, and we see that for other potentials, median value of cognate epitope rank is from 32 to 54%. and this is significantly worse than TCRen, and in a lot of cases this performance is close to performance of a random classifier. This is another representation of this benchmark. Raw curves for distinction between all 91 structures with cognate epitopes and 91,000 structures with unrelated peptides, and we see that TCRen displays accurate distinction between these sets with area under curve for 0.93, while for other pairwise potentials, AUC is close to 0.5. Further, we compared performance of TCRen with Rosetta scoring function and MMPBC approach. These approaches require atomic level representation and calculate energy of interaction on this level, so we can't directly compare them with TCRen, which is residue level. To do this before and atomic level calculations are sensitive to small inaccuracies, so they require refinement or construction of some ensemble of structures. To perform this comparison, we did molecular dynamic simulations of TCR peptide MHC complexes of less 100 nanoseconds. Then we split these trajectories to 1000 frames. For each frame, we calculated energy of interaction of peptide and TCR using either TCRen or Rosetta or MMPBCA, and then average this value across all the frames. As this procedure is computationally expensive, we applied it only to DMR5 model system. For this TCR, a number of peptides recognized are identified using ECCB screening, and these peptides are clustered to two groups, with hydrophobic GE motif and with charged DRG motif. And we perform molecular dynamic simulations for TCR peptide MHC complexes with confirmed GIG epitopes, confirmed DRG epitopes, and unrelated peptides. And we see here that TCRen was able to find significant difference between both GIG peptides and unrelated peptides, and between DRG peptides and unrelated peptides. So it explains recognition of both GIG and DRG motifs. But for Rosetta MMPBC approach, neither of them can do that, and no significant difference between recognized and non-recognized peptides are detected using these approaches. To demonstrate practical applications of TCRen, we demonstrate its use for prediction of cancer neoepitopes recognized by tumor-infiltrating lymphocytes. High mutational load is a hallmark of cancer, and peptides from mutated proteins are presented on the surface of cancer cell by MHC proteins, and these mutated peptides may be detected as foreign by T cells, by tumor-infiltrating lymphocytes, and these tumor-infiltrating lymphocytes may mediate anti-tumor immunity. The problem is that not all neoepitopes can elicit such a response, and we need to predict which neoepitopes are monogenic if we want to use them in cancer vaccines. And here we propose a pipeline utilizing TCRen for this task. From exome sequencing, mutations somatic and non-synonymous in cancer cells are identified, and then peptides containing these mutations and binding to MHC of this patient are considered as candidate neoepitopes. Also, from tumor tissue, tumor-infiltrated lymphocytes may be isolated, and their TCRs may be sequenced. The question is what neoepitopes are recognized by these TCRs. And here what we propose, we do homology modeling of TCR peptide chemistry complex of TCR from tumor-infiltrating lymphocytes with some random peptide. Then we mutate this random peptide for each of candidate neoepitopes, assess estimate edge of interaction of these candidate neoepitopes and TCRs, and then select top peptides for experimental testing. We validate this approach on data from literature, and we see that for this TCR, for which neoepitope was estimated by experimentally, we also predict this neoepitope as #1 in ranked list of candidate neoepitopes. And here are conclusions of my work. Thank you for your attention.